we are still left over with few concepts from cell the unit of life. If we have to cover double membrane bound organelles, microbodies and cytoskeleton, that ends the chapter. So, what we will do today is we will finish off double membrane bound organelles and then we will sort out few of the MCQs, previous year NEET questions, and tomorrow we will solve whole of the NEET MCQs with the revision of cell the unit of life. So, starting with double membrane bound organelles in this chapter, we have 3 double membrane bound organelles to learn. What are they? Mitochondria, plastids and then nucleus. First let us look into mitochondria. Look at the structure of mitochondria. Mitochondria structure varies from cylindrical to sausage shape, and the number of mitochondria present in each cell depends on the type of the function that cell is performing. What is its shape? It can be cylindrical or sausage shape with an approximate diameter of 0.2 micrometer to 0.4 micrometer in diameter and 0.4 micrometer to 1 micrometer in length. An approximate diameter and length of the mitochondria. Look at the components of mitochondria. Yeah, if you can see here, it has outer membrane and inner membrane. In between that we have intermembrane space. If we have to take a small compartment or a box like this, let us suppose this as outer mitochondrial membrane, and next inner mitochondrial membrane which has infoldings into cristae. The space between outer and inner mitochondrial membrane is addressed as what? Intermembrane space. All the enzymes or carriers which are required for aerobic respiration. We know mitochondria is powerhouse of the cell and it is the site where aerobic respiration takes place, right? And all the enzymes and carriers required for that are present both in the cristae and outer mitochondrial membrane. And this cristae is compartmentalizing this mitochondria. This complete, if we assume like an aqueous environment, that aqueous environment is separated by what? Cristae, okay, into intermembrane space and mitochondrial matrix, right? It has intermembrane space and mitochondrial matrix. And we have to know the sites or locations of the reactions that are happening. We know mitochondria is the site for aerobic respiration, and aerobic respiration has how many steps? If we have to recall something from respiration in plants, in higher plants, first step is glycolysis. That is a common step both in prokaryotes and eukaryotes, which takes place in cytoplasm of the cell, right? And rest 3 steps of only aerobic respiration, which includes oxidative decarboxylation, tricarboxylic acid cycle and electron transport chain or electron transport system. These are the 3 steps of aerobic respiration happens in mitochondria. Let us recall on that. And what are the components of mitochondrial matrix? Mitochondrial matrix consists of 70S ribosomes or 80S ribosomes? 70S ribosomes, very good. 70S ribosomes, and it consists of double stranded circular DNA, RNA and proteins. These are the components of what? Mitochondrial matrix. And can you see any similarity of mitochondria with prokaryotic cell here? Can you see any similarity of mitochondria with prokaryotic cell? What are they? 70S ribosomes, you find it in prokaryotic cell as well, and double stranded circular DNA. So, there is a concept that we learned under evolution, endosymbiotic theory. Do you remember? Endosymbiotic theory. According to this theory, both mitochondria and chloroplast are in the ancestral time, at the origin of evolution, both mitochondria and chloroplast are individual prokaryotic cells. In due course of evolution, eukaryotic cells, when they are evolved, they have engulfed these prokaryotic cells, and mitochondria and chloroplast have become what? Part and parcel of eukaryotic cells, right? But then they have the characteristics similar to the prokaryotic cell. That is the reason. Did you observe both mitochondria and chloroplast can synthesize their own ATP? Energy synthesis happen. Though mitochondria is called as powerhouse of the cell, chloroplast is addressed as kitchen of the cell. Both the organelles can synthesize their own energy, their own ATP. It is because they are individual cells long ago, and in due course of evolution, they have entered into eukaryotes by endocytosis, and they have become part and parcel of eukaryotic cells. Understood, right? So, you have to remember this endosymbiotic theory. You have to remember the components or constituents of mitochondrial matrix. Got it right, ma? Clear with mitochondria? Yeah. Fine. Now, let us look into the sites or actions of aerobic respiration, sites or locations of aerobic respiration. Let us remember the gist of the cycles or the products of each step in aerobic respiration. Assume this as a cell and mitochondria inside the cell. Tell me the compartments. This is outer mitochondrial membrane, right? And this is inner mitochondrial membrane. This will be intermembrane space and mitochondrial matrix, right? So, here if you see the first step of respiration, what is that? Glycolysis. So, we all know that glycolysis is a 10-step process, begins with glucose, and the product of glycolysis is pyruvate, right? Two molecules of pyruvic acid are formed. These two molecules of pyruvic acid, they will directly land into the matrix of mitochondria. Okay. So, first step happening where, ma? Inside the cytoplasm. Second step will happen in mitochondrial matrix, that is TCA cycle or Krebs cycle. Sorry. Second step is oxidative decarboxylation. Pyruvic acid will undergo the second step, that is oxidative decarboxylation. In oxidative decarboxylation, what happens? Do you remember? Pyruvic acid will turn to acetyl-CoA. And this acetyl-CoA, which is the starter of TCA cycle, here the journey of TCA cycle will start again where? Inside the mitochondrial matrix itself. Got it? Second is TCA cycle or Krebs cycle. Right, ma? Wherein acetyl-CoA will start continuing this cycle, and at the end of this cycle, this will synthesize high energy rich molecules NADH, FADH2 and ATP. How much amount of NADH, FADH2, that we will learn in bioenergetics of TCA cycle when we are discussing respiration in higher plants. As of now, you remember the site of the reaction. Okay? Where is TCA cycle happening? Inside the mitochondrial matrix. And last step of aerobic respiration will be ETS cycle, electron transport system or electron transport chain. This happens in inner mitochondrial membrane. There are 4 complexes. I told you, right? All the electron carriers and protein channels involved in aerobic respiration, where are they present beta? They are present in inner mitochondrial membrane. And all the enzymes required for the respiration are separate. Based on their function, they are present both in inner mitochondrial membrane and outer mitochondrial membrane. But then electron carriers and protein channels are embedded in inner mitochondrial membrane itself. It consists of what? Electron carriers and protein channels. Likewise, for ETS to happen, we need 4 complexes, if you remember. First complex is NADH dehydrogenase, second succinate dehydrogenase, third cytochrome BC complex, fourth cytochrome C oxidase complex. Likewise, we have 4 complexes which are present where? In inner mitochondrial membrane itself. So, ETS or electron transport system happens where? In inner mitochondrial membrane. Or cristae also you can write it as. And cristae will also help in what? Increasing the surface area of the mitochondria, wherein all the reactions can happen freely. Okay. So, I hope you are clear with the structure of mitochondria and functions or sites of all the reactions that happen in aerobic respiration. Clear, right ma? About energetic point of view and mechanism, all the steps we will be learning in respiration chapter. Here though you have to remember the sites. And now we have discussed a few similarities. If you are taking down, please take 2 minutes. Yeah, done. So, if you are discussing the similarities of mitochondria with a prokaryotic cell, here time is not sufficient, you need still more time. Okay, I will save this anyhow, fine ma? Yeah. If we have to discuss the similarities between prokaryotic cell and mitochondria, we have 70S ribosomes in common, double stranded circular DNA in common, and then it comes to the division. How mitochondria will divide? Via binary fission. Division is by binary fission. Clear on everything with mitochondria, no? On its structure, components present inside it, sites and locations of aerobic respiration. If you are pretty confident with this, we will move on to the plastids, right? Yep. So, one more point here to discuss with mitochondria. Here itself we will be discussing, so that in plant physiology it will be easy for us. We have complex 5 which is named as ATP synthase complex. Okay, ATP synthase or F0F1 particles or F0F1 complex. So, after ETS process or electron transport chain, what happens ma? Intermembrane space will be completely equipped with protons. Intermembrane space will be equipped with what? Protons. So, high amount of protons you will be seeing where? In intermembrane space, and low proton concentration will be there in mitochondrial matrix. So, here there is a gradient that is generated, proton gradient that is generated, which activates complex 5. What is complex 5? F0F1 complex. Okay, wherein F0 is embedded inside the inner mitochondrial membrane, F1 is projected towards the mitochondrial matrix. This projection is very important now, because this F0F1 complex will be different in chloroplast and mitochondria. On a comparative analysis might be diagram based, statement based, any way the question can be formed. Okay, F0F1 complex location you have to remember. Okay, should I repeat it once again? Listen carefully. After electron transport chain, what happens? Due to this process, high amount of protons are accumulated in which region? Intermembrane space. High amount of protons are accumulated, and proton reduction will happen where? In mitochondrial matrix, and this generates what? A proton gradient is generated, because of which a pump is activated, that is F0F1 pump. Okay, there are different names for this F0 F1 complex. We can also call it as F0 F1 complex or Fernandez-Morgan particles, or you can also call it as oxysomes or ATP synthase. Right, these are different synonyms of F0 F1 particles. Now, because of this proton gradient, what happens? This ATP synthase will be activated, which has two components in it. F0 is embedded inside the inner mitochondrial membrane, F1 is projected towards the matrix, whereby in a single run 4 protons are pumped and it will generate or it will synthesize 1 ATP. Okay? Clear now? So, it has F0 embedded inside inner mitochondrial membrane, F1 is projected towards the matrix of mitochondria. This position is very important because this will be different in chloroplast again. Okay, chloroplast will be learning chemi-osmotic hypothesis, right? There the position of F0 F1 complex will be different. So, you are clear on it, right ma? Everybody? And now we will be moving to plastids. We have 3 types of plastids, if you remember. 3 types of plastids. What are they? Yeah, chloroplast, chromoplast and leucoplast. Plastids are of 3 types, which includes chloroplast, chromoplast and leucoplast. First we will learn complete structure and functions of chloroplast and then we will move on to chromoplast. Kya hua Toshita? You are fine, no? Yeah. First with chloroplast. Chloroplast has 2 membranes, outer membrane and inner membrane. It is a double membrane bound organelle and the space between outer membrane and inner membrane is addressed as periplastidial space. Space between outer and inner membrane is known as what beta? Periplastidial space, which is around 10 to 20 micrometers in diameter. And here if you look at the complete matrix of mitochondria, matrix of chloroplast, I am still in mitochondria, let us come to chloroplast. The matrix of chloroplast is addressed as what? Stroma. Okay? Matrix of chloroplast is what? Stroma. Matrix of chloroplast, which is a fluid filled space. Okay. Let us discuss about granum later. Let us see what are the components that are present in the stroma. What are the components? It also has 70S ribosomes, double stranded circular DNA, consists of few RNA molecules. It also consists of lipid droplets and starch granules. Right. These are the components of what? Matrix of chloroplast, that is stroma, which is fluid filled space inside the chloroplast. Chloroplast is already double membrane bound, we know that. Thylakoids are membranous sac-like structures which are like third membrane system of chloroplast. They form what? Third membrane system of chloroplast. They are membranous sac-like structures. Membranous sac-like structures, right. So, this membranous sac-like structures, it also has a membrane. This membrane is addressed as thylakoid membrane or thylakoid lamellae. This complete membrane is known as what? Thylakoid membrane or thylakoid lamellae. And the space inside this thylakoid is known as what? Lumen of the thylakoid. This is known as thylakoid lumen. First you listen ma, then you can read. If not, it will be a kichdi. Okay. So this is what thylakoid membrane, which is third membrane system of chloroplast, and this is thylakoid lumen, which is the space present inside the thylakoid. Now here you got, and this stalk of sac like structures. Thylakoids are membranous sac like structures. If they are present one above the other, they will make up what? Stalk, like pile of coins. That stalk of thylakoids is known as what? Granum. Stalk of thylakoid is what? Granum. One granum is connected to other granum by stroma lamellae. This is the structure. One granum connected to other granum by stroma lamellae. Clear, right? So once again let us quickly revise. Plastids are of three types: chloroplast, chromoplast, leucoplast. Chloroplast, if we see, it is double membrane bound. Space between two membranes is known as periplastidial space, and inside that fluid filled space is known as stroma, which is addressed as what beta? Matrix of chloroplast. It includes the components like 70S ribosomes, double stranded circular DNA, RNA, lipid droplets and starch granules. One more point to tell you. Though chloroplast and mitochondria are evolved from prokaryotes under endosymbiotic theory, these organelles are also known as semi-autonomous cell organelles or self-duplicating organelles. Okay, because they can replicate their own DNA. Okay, remember that. Semi-autonomous or self-duplicating organelles. And these three features you remember, right? What are the similarities with prokaryotes, ma? 70S ribosomes, double stranded circular DNA, binary fission. These three are the similarities with it. And then next, when it comes to chromoplast, with chloroplast everything is clear, right? After learning chromo and leucoplast, like how we have learned sites of reactions, same way we learn the sites of light and dark reactions happening in the chloroplast. Okay, now chromoplast. Chromoplast emit different colors because of the pigments that are present in it. It can be anthocyanins, it can be carotenoids, xanthophylls, any of the colored pigments that are present in chromoplast. It emits what? Color. Okay. And next is what? Leucoplast. Leucoplast is of three types based on the storage material that it is storing. What are the three types? Can you name? Amyloplast, aleuroplast, ileoplast. Amyloplast will store what? Carbs, carbohydrates will be stored. Aleuroplast are the bodies which will store proteins, they are protein granules. Ileoplast will store what? Fat globules or lipid droplets. These are three types of leucoplast and they are non-colored. Based on the requirement or necessity of the cells, leucoplast can convert to what? Chloroplast again. Interconversion is possible. Leucoplast can be interconverted to chloroplast based on the necessity of the plant. Okay. Now let us look into the chloroplast sites of the reactions. And when it comes to the shapes of the cells, chloroplast can be of any shape. We have seen chloroplasts are present in euglenoids, which are single celled protists. All plant type of protists, chloroplast is present in them. Algal cells, chloroplast is present. Higher plants, chloroplast is present. So chloroplast is present in all type of organisms, unicellular to multicellular. If it is performing photosynthesis, so it has chloroplast. Here you see they are of different shapes: cup shaped, discoid, C shaped, ribbed, reticulate, spiral and stellate. These are different shapes of the chloroplast. What you have to remember is this table. Not completely, I will tell you what exactly you have to remember. Cup shaped chloroplast you will see in Chlamydomonas, which is unicellular green algae. Okay. Chlamydomonas, what type of chloroplast will be there? Cup shaped chloroplast. Discoid shaped chloroplast you will see in Chlorella. Chlorella is again unicellular green algae. Then C-shaped chloroplast you will see in Eulothrix, which is filamentous green algae. This you need not to remember, Edogonium is not an example for you. Reticulate, again you have to remember Cladophora, where this is used by E.W. Engelmann for the experiment. We have learnt in plant physiology. So you have to remember this. Reticulate shaped chloroplast is present in Cladophora. Spiral shaped chloroplast is present in Spirogyra, and then stellate shaped in Zygnema. This also you can ignore. Except these two examples you have to remember all. Okay. Cup shaped you can see it in Chlamydomonas. Be louder so that you can remember. Discoid, C shaped, reticulate, spiral, right. These are different shapes of the chloroplast. Now let us look into the sites of the chloroplast reactions or sites of light and dark reactions. We know photosynthesis occurs in two reactions. What are they? Light dependent. Yeah. Light dependent and light independent reactions. So light dependent reactions include, first is what ma? Absorption of light in the form of photons. This happens where? In thylakoids, because here we have learnt thylakoid membrane, right? If you remember, thylakoid membrane will have what? Photosystems, which are light harvesting complexes. Do you remember it has what? Photosystems. These are light harvesting complexes. This light harvesting complexes consists of two types of pigments, core pigments and accessory or antenna pigments. Remembered? Third membrane system of chloroplast is what? Thylakoid. The membrane name is what? Thylakoid membrane or lamellae. It consists of what? Light harvesting complexes named as photosystem 2 and photosystem 1. They consist of pigments. They are classified into two types, core pigments and accessory or antenna pigments. Core pigment includes what beta? Chlorophyll A. Core pigments include chlorophyll A. Accessory pigments include chlorophyll B, carotenoids and xanthophylls. Okay, consists of chlorophyll B, carotenoids and xanthophylls, right. They will absorb light at different wavelengths. That and all we will discuss in the photosynthesis class. As of here though, what you have to remember? Thylakoid membrane consists of light harvesting complexes which has two sets of pigments and they will absorb light at different wavelengths. So absorption of light in the form of photons takes place where exactly? Thylakoid membrane. Got it right, ma? This happens in thylakoid membrane. And second reaction is what? Photolysis of water. Photolysis of water. Drink some water. Take down parallelly. Photolysis of water takes place where? Thylakoid lumen. Okay. This takes place in thylakoid lumen. And third reaction under light dependent reactions is formation of assimilatory power, that is ATP and NADPH. Where are they formed? In the stroma of the chloroplast. Now coming to light independent reactions, the only reaction that happens here is taking the energy, that is assimilatory power, which is present in the stroma. It forms what? Glucose formation or glucose fixation takes place. carbon dioxide fixation or glucose formation. So here glucose formation which takes place where, again in the stroma of the chloroplast. This can happen in many cycles. We will be learning C3, C4, CAM on different plants, right? Clear? Yeah. So this is all about chloroplast or plastids. Taking down. Done? Yeah. And then different types of plastids are done, and now we are entering to the core organelle, that is nucleus. Okay. So here from last 2 classes also we have discussed what are the components of nucleus. Okay. And we have learned about enucleated cells. Can you name few enucleated cells that we have discussed? Huh? Human RBC, tracheids, sieve tube cells, all these are enucleated. Can you tell me any xenocytic cells? Rhizopus, opalina. Opalina is also xenocytic. It has hundreds of nuclei inside it. Okay, these are what? Multinucleated cells. We have discussed about a protozoan, which is a ciliated protozoan of binucleated condition. Can you tell me? Louder? Very good. Paramecium. Ciliated protozoan, paramecium, it has 2 nuclei, binucleated condition. Okay, wherein large nucleus will take hold of the metabolic activities of the cell, and small nucleus, that is micronucleus, will take care of reproduction of the cell. We have discussed, right? Recalled everything? Now, let us look into the components of the nucleus. There are 4 components of the nucleus. First, nucleolus. It can be one or more spherical bodies inside the nucleus. First component, nucleolus, can be one or few, and its components will be continuous with the cytoplasm, and nucleolus is the region where rRNA synthesis takes place by RNA polymerase 1. RNA polymerase 1 will synthesize what? rRNA, ribosomal RNA. Okay. And then second component is what, ma? Nucleoplasm. Nucleoplasm will consist of genetic material in the form of chromatin. Okay. It consists of the genetic material in the form of chromatin. And it is the region where tRNA and mRNA are synthesized. mRNA is synthesized by what RNA polymerase? If you remember, RNA polymerase 2 will synthesize mRNA. RNA polymerase 3 will synthesize what? tRNA, which is also known as soluble RNA. These sites and locations you have to remember. Okay, might be the questions are asked from molecular basis, but the regions and locations here itself, if you remember, it will be easy for you people there. Next, so nucleolus, nucleoplasm, then is nuclear pore. Third region is what? Nuclear pores. This nuclear pores will enter as the transport channels for the transport of or influx and efflux of RNA and protein molecules, okay, into the nucleus and cytoplasm. These are like gates to transport the molecules in and out. What molecules? RNA and proteins. Next is what? Nuclear envelope, which will surround what? Nuclear membrane. Like that we have outer nuclear membrane and inner nuclear membrane. Okay, outer nuclear membrane and inner nuclear membrane. Fine. So, how can we observe this mitochondria and nucleus? These organelles, they are visible only under electron microscopes when they are densely stained, right? So, you are clear on the components of nucleus, right? Nucleolus, nucleoplasm, nuclear pores and nuclear envelope. And then now we have to learn something on the genetic material or on the structure of the chromosomes might be, shapes of the chromosomes might be. We have learnt that it will be in the form of chromatin, right? But if at all, if you see in your state textbooks and all about condensation of chromosomes, it is given here only. So, what is wrong? We will learn it here, condensation of chromosomes. How chromatin is condensed to chromosomes. Condensation of chromatin to chromosomes. You tell me, chromatin consists of what? DNA. This is what? Completely DNA. DNA is negatively charged or positively charged? Negatively charged. So, to condense negatively charged DNA, it has to be associated with positively charged proteins or the basic proteins like called as histone proteins. Okay. So, the negatively charged DNA is associated with the positively charged or basic proteins known as what? Histone proteins. Can you tell me these histone proteins are rich in what amino acids? Histone proteins are rich in what amino acids? I can hear that. Toshida, be louder, you are right. Lysine and arginine. Very good. Lysine and arginine. Good. Okay. So, it consists of these amino acids. When chromatin is binded with histone proteins, what type of histone proteins are binded with this? See, this long chromatin structure is arranged in the form of beads. When it is observed under microscope, it can be like beads on the string. Okay, how histone protein, how DNA is wrapped around this histone proteins, like it appears like what? Beads on the string. What type of histone proteins are present? H2A, H2B, H3, H4. One set of these 4 proteins are present in how many sets? 2 sets. So, total how many proteins are present in a single nucleosome? 8 histone proteins. Okay. When DNA is wrapped around this histone octamer, as these are 8 histone proteins, can I call them as histone octamer? Yes, right? This is a histone octamer. When DNA is wrapped around this histone octamer, okay, if you see that particular region, that is addressed as what? Nucleosome. Can you see here? This region is nucleosome. How much length of DNA is wrapped around one histone octamer, or how much length of DNA is present in one nucleosome? It is 200 base pairs of DNA. Is present in one nucleosome. Fine, right? There should be one DNA that will be connecting between one nucleosome to another nucleosome. That DNA is addressed as linker DNA. Okay, this is known as what, ma? Linker DNA. Okay, once again shall we quickly recall this? Condensation of chromatin to chromosomes. This entire process happens inside the nucleoplasm. Exactly where? Nucleoplasm. DNA is negatively charged. So, to condense this, the small logic, it requires what? Positively charged or basic proteins known as what, ma? Histone proteins. These histone proteins are rich in what amino acids? Lysine and arginine. What are the proteins that are present? H2A, H2B, H3, H4. How many sets of them? 2 sets. So, this is addressed as what? Histone octamer. Okay, histone octamer. And this forms what? One nucleosome. When DNA is wrapped around this histone octamer, how much length of DNA is wrapped? 200 base pairs of DNA for one nucleosome. What is the necessity of the condensation actually? Why chromatin has to be condensed? What is the necessity of the condensation? If you remember cell cycle, that in S phase what happens? DNA will duplicate. DNA duplicates in S phase, wherein DNA is duplicated but inside the same cell, inside the same nucleus. Okay. So, chromatin condensation is mandatory to fix that long DNA. How much DNA? Approximately in 48 chromosomes, 46 chromosomes, you will be having how much amount of DNA? 2 meters of DNA, 2 meters long DNA has to fit in that small 10 to the power of minus 6 meters nucleus, right. So, to fix it inside the nucleus, condensation of DNA is mandatory. Condensation of chromatin is mandatory. That happens via histone proteins. What are those basic proteins? What are the amino acids present inside it? How much amount of DNA can you see in a single nucleosome? You have to remember. Clear, right? Clear? Everybody? Yeah. This is about condensation. Now, we have to learn on structure of the chromosome. Okay. It is not given, just we will learn it from the metacentric chromosome itself, or else, yeah. First we will see structure. Can you name or recall the phases of karyokinesis? Sit straight everyone. Recall the phases of karyokinesis. Prophase, metaphase, anaphase, telophase. Amongst these 4 phases, tell me at which phase you can see clear structure of chromosome? Structure of chromosome. Metaphase. At the end of prophase and in metaphase, the complete condensation of chromatin will occur and you can see shortest and thickest chromosomes. Okay. So, structure of chromosome is clearly observed in metaphase. So, if I have to take the chromosome structure here, okay, this complete thread or strand is known as a single chromatid. Okay. So, how many chromatids you have here? 2 chromatids, 1 and 2, 2 chromatids, and these are known as arms of the chromatids. Okay. The arms of the chromatids, if you see in shapes of chromosomes, okay, one arm is short and one arm is long, short and long arm. Short arm is addressed as P arm and long arm is addressed as Q arm. Okay, P and Q arms. And here you see these are 2 chromatids. Each chromatid has 2 arms. If the length is equal, you are not naming it. If one arm is short and other arm is long, then you are naming it as P arm and Q arm. And at the center, the chromatid is holded by a constriction. This is known as what, ma? Primary constriction. Primary constriction, also known as what? Centromere. To the primary constriction or centromere, you have small disc shaped structures like this. Small disc shaped structures. These disc shaped structures are known as what? Kinetochores. These disc shaped structures are kinetochores. And there is this complete set of chromosome has what? Genomic DNA. But at the time of density gradient centrifugation, when you are centrifuging the chromosomes or nucleus, you can see small repetitive DNA sequences which are coding apart from the genomic DNA. That repetitive sequences are known as satellite DNA. Okay, satellite DNA which consists of repetitive DNA, satellite DNA sequences which consists of AT and GC sequences. Okay, this will form satellite DNA or secondary constriction of the chromosome. Satellite DNA or secondary constriction of the chromosome, right. So, this satellite DNA or secondary constriction will help in what? It will help to maintain the chromosome structure at the time of cell division or karyokinesis. Chromosomes will be duplicated, right. Centromere will be duplicated, right. So, at this point of time it will help to maintain the chromosome structure without erasing the ends of the chromosomes, right. And there are many other benefits of satellite DNA. As of now, you remember only this one thing. Satellite DNA will help in maintaining chromosome structure, stability, right. So, let us recall quickly. Structure of chromosome is clearly seen in metaphase. Primary constriction, also known as centromere, which will hold two arms of the chromatids. Likewise, we have one and two arms in this chromosome, and you have few disc shaped structures, two disc shaped structures along the primary constriction. What are they? Kinetochores. Okay, and here we have satellite DNA or secondary constriction which has DNA repetitive sequences. Clear till here, right? Now, shapes of the chromosomes. Shapes of the chromosomes are clear at which phase? Sangeeta is too serious today. Shapes of the chromosomes are clearly visible in which phase? Anaphase. Good. Anaphase, right. Like after centromere splitting, chromatids will be moving to the opposite poles. Do you remember that in anaphase? Centromere splitting and chromatids move to the opposite poles. If two arms of the chromatids are equal, you will call it as V-shaped chromosomes or metacentric chromosomes. V-shaped or metacentric. If one arm is short and other arm is long, this is known as what? L-shaped chromosomes, and this is known as submetacentric. Okay, one very short arm and one very long arm is known as what? Acrocentric chromosome. What shape? Very good. J shape. And one extremely short and negligible arm, you cannot see, one arm is not visible, and other arm is very long, that is known as what? Telocentric. And what shaped? I shaped. Okay, V, L, J and I shaped. Short arm is known as what? Long arm? That term is not asked till now, but keep it in your mind beta. Like if they have one short arm and long arm, those are known as heterobrachial chromosomes. Heterobrachial. So, clear on structure and shapes. Condensation of chromosome, components of nucleus, where rRNA is synthesized? rRNA is synthesized in? Louder. rRNA is synthesized in nucleolus. tRNA and mRNA? Nucleoplasm. rRNA is synthesized by RNA polymerase 1, mRNA by RNA polymerase 2, tRNA by RNA polymerase 3. Clear till here? Mitochondria, chloroplast, chromo, leuco, nucleus, condensation of chromatin, structure of chromosomes, shapes of chromosomes. These are the concepts we have drilled today. Okay. Now, let us see few of the PYQs. Ready to solve? What's going on there? Tomorrow. Exhausted, I can see. So till here it is fine, right? Yeah. Now let us practice the NEET PYQs first. So first we will start with re-NEET. We will see how much you have answered, how much you can recall and what you can do now. We will see, okay. We have equal numbers, so first chance we will give it for boys. Yeah, you 3 people. I need explanation also with answer. So take your time. Students from virtual platform also can answer. Yep. Aypoyinda Sangeeta? Relax. Frame the explanation. Drink water all. Have a sip of water. It's been close to an hour, right? Okay. Done? Okay. Turn for you. Girls? 4? Okay. So, let us see it once. Select the correct statements regarding the cell membrane in eukaryotes. Membrane synthesis includes Golgi complex, endoplasmic reticulum and mitochondria. In endomembrane system we have learnt ER will synthesize proteins and lipids. Their modification is done in Golgi bodies, and glycoproteins, glycolipids are synthesized from ER and Golgi bodies. But nowhere mitochondria is present in endomembrane system, or nowhere it is involved in membrane formation. So this is wrong. Statement 2 we will observe. Ribosome on endoplasmic reticulum bears ribosomal surface. Both mitochondria and chloroplast has circular DNA, just now we learned. Because learnt under what theory? Learnt under what theory? Endosymbiotic theory. Remember that term, yaar. Third, so this is done. A network of microtubules, microfilaments and intermediary filaments present in cytoplasm is called cytoskeleton, correct? We will learn about 3 in detail tomorrow. Next, mitochondria is single membrane bound organelle? No. So what is the right answer? B, C, D. One small logic. Immediately looking at the statement, you can eliminate this narrow. Okay, fine. Yeah, next question. This you see. Same, I have picked this from re-NEET only. Around 6 to 7 questions only from re-NEET, I am telling you. From this chapter only. Non-membrane bound organelles found in both prokaryotes and eukaryotes. Ribosomes. In prokaryotes, what type of ribosomes you can see? Where are they present? Attached to plasma membrane and few in cytoplasm. Okay, those ribosomes attached to plasma membrane will help in translocation of proteins, and this is purely involved in protein synthesis. And you will see one condition at the time of translation. Very good, Skanda. That is what? Polysome condition. This is how you have to recall and practice. One term given, entire history you have to draw out. Next, in eukaryotes, what type of ribosomes you will see? ATS, 70S. ATS where do you see? Attached to endoplasmic reticulum and present inside the cytoplasm. 70S ribosomes where do you see? Very good. Chloroplast and mitochondria. Done? Yeah, this is about ribosomes. Next, this question. Nucleolus. tRNA, mRNA, nucleoplasm. What is the RNA polymerase involved? RNA polymerase 1. Next question. Of course, this is from respiration also, but then it is connected to mitochondria structure, so I have picked this. Very good. Keep answering. Four. Good, Sangeeta. We'll see. Glycolysis. Oh, option 4 ఇయ్యలేదంటున్నారా? Fine. Cytoplasm. It's given, no, right? Just a second. See, glycolysis happens where? Inside cytoplasm. Electron transport system operates in inner mitochondrial membrane also, you can say. Or cristae. Right. Accumulation of protons in intermembrane space. Krebs cycle in mitochondrial matrix. Recalled? Next. Somewhere this option is missed, but now it is fine, right? Yeah. Next question. On condensation of chromosomes. A? We'll see. Any other options? Yashwant said A. B? What is your option? You are agreeing with this or you are giving any other option? Okay, think. 2 more minutes. There should be some paper, pen to decode. You people are superhumans. Just like this on air, you people will be saying. Second? Okay, we'll see. Which of the following statements are correct with reference to packaging of DNA? Histones are organized to unit of 8 molecules called histone octamers. Histones are positively charged basic proteins. Histones are rich in basic amino acids like lysine and arginine. Positively charged DNA, wrong. DNA is negatively charged. Packaging of chromatin at higher level requires additional set of proteins known as non-histone chromosomal proteins. Correct actually. So A, B, C and E. Option లేకపోతే ఉన్నది తీసుకోవాలి. A, C, E. Right. Next. NEET 2025. పర్వాలేదు చూద్దాం. One point to be added. You solve this question, I will add it. 3. See, still thinking. The eukaryotic ribosomes are 80S and prokaryotic ribosomes are 70S. Each ribosome has 2 subunits. The 2 subunits of 80S 60 and 40. 70S is 30, 50 and 30. This is wrong again because it is not 20. It is also not 20. 80S is not 60 and 30. So, what is the right answer? A, B, C. Okay. Next. This question you see, ma. So, here we have learnt what are the components of chloroplast, mitochondria and all. You should know what are the constituents of a chromosome also. Chromosome after condensation, it consists of DNA, it consists of RNA, it consists of histone proteins and non-histone proteins also. These 4 are the components of a chromosome. Clear? Remember that. And now we are with this question. 2. We will see. Centromere, cell division. Cilium, cell movement. Cristae present in mitochondria. Cell membrane has phospholipid bilayer. Next, this one. Lysosomes. Last questions of today's session. Specialized membranous structures. Histones are rich in lysine and arginine. Okay. So tomorrow we will wind up the cell the unit of life, and we will practice all NEET PYQs. I will pick some from EAP set. I will pick some statement based. We will have a good objective session tomorrow. So come prepared. Okay. Yeah. Thank you.